modern technology is revealing more and more about our genetic makeup.
How does genetics point to design? This week on Creation Magazine Live.
Welcome to Creation Magazine Live. My name is Richard Fangrad.
And I'm Calvin Smith.
Now, this week on Creation Magazine Live, our topic is how does genetics point to design?
Right. Well, the field of genetics and evolution, they've kind of been in conflict. They've been enemies since the beginning of both concepts. Gregor Mendel, the father of genetics, and Charles Darwin, the father of modern evolution, were contemporaries. And at the same time that Darwin was claiming that creatures could change into other creatures, Mendel was showing that even individual characteristics remain constant.
Right. Yeah, well, Darwin's ideas were based on erroneous and untested ideas about inheritance. Mendel's conclusions were based on experimentation. Only by ignoring the total implications of modern genetics has it been possible to maintain the fiction of evolution. Now, to help us understand biology based on creation rather than evolution, let's sample some of the evidence from genetics arranged under four supposed sources of variation, which are environment, recombination, mutation, and creation. We'll have a look at those four.
Okay, well, let's start with environment. This refers to all of the external factors which influence a creature during its lifetime. For example, one person may have darker skin than another simply because, they're exposed to more sunshine, for example.
Okay, all right. Another may have larger muscles because he exercises more. Mm-hmm. Such environmental caused variations generally have no importance to the history of life because, of course, they cease to exist when their owners die.
Right.
Those traits aren't passed on.
Yeah, in the middle 1800s, some scientists, like Lamarck, for example, believed that variations caused by the environment could be inherited. And Charles Darwin accepted this fallacy, and it no doubt made it easier for him to believe that one creature could change into another.
Right. He thus explained the origin of the giraffe's long neck, in part through the inherited effects of increased use of parts, he said. In seasons of limited food supply, Darwin reasoned that giraffes would stretch their necks and stretch for the high leaves, supposedly resulting in longer necks being passed on to their offspring. And now we know, of course, that doesn't work at all.
Right. Well, let's look at recombination. This basically involves shuffling genes and is the reason that children resemble their parents very closely, but not exactly like, one another. So the discovery of the principles of recombination was Gregor Mendel's great contribution to the science of genetics.
Yes.
Mendel showed that while traits might be hidden for a generation, they were not usually lost, and when what seemed to be new traits appeared, it was because their genetic factors had been there all along.
Right. Recombination makes it possible for there to be limited variation within the created kinds. But it's limited because virtually all of the variations are produced by a reshuffling of the genes that are already there.
Yeah. For example, from 1800, plant breeders tried to increase the sugar content of the sugar beet, and they were very successful. Over some 75 years of selective breeding, it was possible to increase the sugar content from 6% to 17%.
Cool.
But there, the improvement stopped, and further selection didn't increase the sugar content. Why? Well, because all of the genes for sugar production had been gathered into a single variety and no further increase was possible.
Right. It's like Darwin's finches he observed on the Galapagos Islands. In this single group, we can see wide variation in the appearance and lifestyle among these birds. Now, Darwin provided what is likely a correct interpretation. Darwin was right about a number of things, of how the finches came to be the way they are. A few individuals, he said, were probably blown to the islands from the mainland, from South American mainland, and today's finches are the descendants of those pioneers.
Absolutely.
Probably what happened.
Yeah. However, while Darwin saw the finches as an example of evolution, we can now recognize they're merely the result of recombination within a single created kind.
Right.
The pioneer finches brought with them enough genetic variability to be sorted into the varieties that we can see today. And that's not evolution, of course, because it doesn't involve the creation of new genetic information. I don't know how many times we need to repeat this. If you're going to turn a microbe into a man, you have to add new genetic information for forms, functions, and features that didn't exist before, not separating genetic information that's already in existence.
And that's what science never shows.
Right.
Yeah.
Right.
Right. So now, when we get back, we'll continue to talk about another source of variation in living things, mutation. And we'll do that when we get back.
Does the human Y chromosome suggest that men are headed for extinction? In 2003, an Oxford University geneticist claimed that the human Y chromosome was crumbling before our very eyes and that the demise of men was imminent. Since this time, other researchers have pointed out that these doomsday predictions were overstated. For instance, the Y chromosome has a unique mechanism for correcting harmful mutations. Nevertheless, the Y chromosome certainly shows signs of overall decay, as do the other chromosomes. Human genetic decay is a real phenomenon, but it flies in the face of evolutionary ideas. According to evolutionists, all the complex coded information in our genomes supposedly arose through a slow accumulation of random changes called mutations. However, what we see with the Y chromosome is that such natural processes consistently degrade the genetic instructions as opposed to create them. Since the time of Adam, we live in a decaying world, just as the Bible says.
To find out more from Creation Ministries International, visit our website, creation.com.
Well, if you just tuned in, this week we're talking about genetics and how it demonstrates design in living things. We've been talking about various mechanisms for variation in living things. And as we said before the break, mutations are next. Let's get into mutations.
Okay, well, first of all, what are mutations, right?
Good place to start.
Yeah.
Let's start there.
Mutations are mistakes in the genetic copying process.
Yes.
Every living cell has intricate molecular machinery designed for accurately copying DNA. So that's the genetic molecule, right? So as this copying process takes place, mistakes do occur. We live in a sin-cursed world. Things don't replicate perfectly.
Yeah.
And even though they happen, they don't happen very often. So once in every 10,000 to 100,000 copies, a gene will contain a mistake, a spelling mistake.
Now, the cell has machinery, amazing machinery, for error correcting these mistakes, but some mutations still slip through. What kinds of changes are produced by mutations? Well, some have no effect at all, or produce a small change, a change that that may have no appreciable effect on the creature, but many mutations have a significant effect on their owners.
Right. Now, based on the creation model, what kind of effect would we expect from random mutations from genetic mistakes? Well, we'd expect virtually all of those, that make a difference to be harmful,
right.
To make the creatures that possess them less successful than before. And this prediction is borne out pretty convincingly. And here are some examples that are going to help illustrate this.
Right. Geneticists began breeding the fruit fly, for example, soon after the turn of the century. And since 1910, when the first mutation was reported, some 3,000 mutations have been identified. All of the mutations are harmful or neutral. None of them produce a more successful fruit fly, exactly as predicted by the creation model.
Right. Now, when we say most of them would be harmful, even the ones that are neutral in these flies or in any organism, as they continue to gather, eventually they will become negative in the sense that you're just degrading the entire bit of information.
The information coded there is being degraded by these mutations, even if it's not borne out in the physical structures or
it's
behavior
like rust
of the
on a
individual.
car. Eventually, it's going to get you.
Yeah.
So, is there then are we saying there's no such thing as a beneficial mutation? Well, yes, there is. A beneficial mutation is simply one that makes it possible for its possessors to contribute more offspring to the future generations than ones that don't have that mutation. So, how does that work? Well, for example, Darwin, he called attention to wingless beetles on an island of the island of Madeira. For if you have a beetle living on a windy island, wings can be a definite disadvantage.
Sure.
Because creatures in flight they're more likely to be born to sea, blown into the sea.
Blown off into the ocean. Yeah. So mutations producing a loss of flight could be beneficial, could be helpful, and the example of sightless cave fish would be a similar example. Eyes are quite vulnerable to injury, So a creature that lives in a pitch dark environment would benefit from mutations that would replace the eye with maybe a scar-like tissue, reducing that vulnerability to injury and disease.
Right. You know, in the world of light, having no eyes, well, that'd be a big handicap, but it's no disadvantage in the dark cave. So while these mutations produce a drastic and beneficial change, it's important to notice that always it involves less genetic information and never a gain. We don't observe the reverse occurring, right? Namely, wings or eyes being produced on creatures which never had the genetic information for them in the first place.
Yeah. So natural selection is the obvious fact that some varieties of creatures are going to be more successful than others. It's pretty basic. And so they will contribute more offspring to future generations. A favorite example of natural selection is the peppered moth in England years ago. As far as anyone knows, the moth has always existed in two basic varieties, speckled and solid black.
Right, and in pre-industrial England, many of the tree trunks were light in color and thus provided a camouflage for the speckled varieties. This is the story, right?
This is the story, yes.
And the birds tended to prey more heavily on the black variety, and then moth collections showed that many more speckled than black ones. And when the industrial age came to England, pollution darkened the tree trunks, so the black variety was hidden and the speckled variety was conspicuous, and soon there were many more black moths than speckled. Now, by the way...
And that was in textbooks for many years as, hey, look, here's evolution happening.
Yeah. And this research that we're talking about here actually is very dubious at best. The actual studies showed that the moths don't even rest on trees during the day, and there's a lot of fabrication going on, but we'll save that for another day.
So, as populations encounter changes in the environment, such as the examples just described, or the result of migration into a new area, that type of thing, natural selection favors the combination of traits which will make the creature more successful in that new environment. This might be considered to be, it might consider to be a positive role of natural selection. The negative role of natural selection is seen in eliminating or minimizing harmful mutations in a population when they occur.
Right. So now, when we get back, we're going to cover one source of variation we haven't talked about yet, and that's, of course, creation, when we return.
Are you skeptical about Christianity? Perhaps you're a Christian but know someone who won't consider Christianity. Christianity for Skeptics is one of CMI's most popular books. Written by Drs. Steve Kumar and Jonathan Sarfati, this powerful resource refutes many attacks on the Christian faith. It contains cutting-edge research, solid theology, and a summary of the Christian roots of science. Questions about Islam, atheism, suffering, evidence for God, and more are answered. Full of bright, catchy illustrations and a sleek modern style, this book draws in any reader. Purchase this resource and many others at creation.com.
All right, welcome back. On this week's episode, we're talking about how does a study of genetics point to design? That's our topic today.
Right. Now, the one source of variation we haven't talked about yet is, of course, creation. And the reason we're going to talk about it is because the first three sources of variation we discussed are woefully inadequate to account for the diversity of life that we see on Earth today.
Right, yeah. Now, an essential feature of the creation model is God having placed considerable genetic variety in each created kind at the beginning. Only then can we explain the possible origin of horses, donkeys, zebras, for example, from the same kind.
Right.
Lions, tigers, leopards from the same kind, and some 118 or more varieties of domestic dogs, as well as the wild dogs, jackals, foxes, wolves, dingoes, coyotes, that type of thing, from the same kind.
Right. You got a whole bunch of information, you split it up, you get different varieties of the same
within
kind.
a kind. All makes sense.
And of course, as each kind obeyed the Creator's command to be fruitful and multiply, the chance processes of recombination and the more purposeful process of natural selection caused each kind to subdivide into the vast array that we see them today.
Right. Yeah, the way to understand variation and speciation is through the removal and rearrangement of genetic information within the original genomes created with an excess of genetic elements. Lots of traits there originally. These genomes initially contained all of the mechanisms required to quickly respond and adapt to changing environments. It's programmed adaptation type of a thing.
Absolutely.
They provided organisms with the tools needed to invade many distinct different niches of environment and so on, and were ideal for the swift colonization of all corners of the world.
Yeah, I mean, these multifunctional genomes, and I think we've used this analogy before, they can kind of be compared to like a Swiss Army knife, right?
Right, yeah.
You know, a Swiss knife contains a lot of different functions, and they're not immediately necessary in a particular environment, but some of them, they're extremely handy in the mountains, other in the woods. Still others maybe for opening bottles and cans or tools for making a fire. So depending on where you are, you may require different sets of functions, and,
Yeah.
animals can be like that too.
In a similar way, depending on where the organism lives, it demands different functions from its genome.
Right.
The environment then determines what part of the non-essential genome is under constraint, and it's only this part that will be conserved.
Right. I mean, if we can crossbreed a zebra and a horse to produce a zorse, a lion and a tiger, a liger or a tigon, or a false killer whale and a dolphin, a wolphin, what does this tell us about the original kinds of animals that God created?
Well, it means that the animals that can hybridize descended from the same created kind. If two animals or two plants can hybridize, they must belong to the same original created kind. If the hybridizing species are from different genera in a family, it suggests that the whole family might have come from one created kind originally. If the genera are in different families within an order, it suggests that maybe the whole order may be derived from an original created kind. We can reason in that particular way.
Right. So God created all kinds or basic types of creatures and plants with the ability to produce a variety in their offspring. And these varieties come from recombinations of the existing genetic information created in the beginning through the marvelous reproductive method created by God. And since the fall, some variations also occurred through degenerative changes caused by mutations.
Right, yeah. The variations allow for the descendants of the created kinds to adapt to different environments and fill the earth as God commanded. It's brilliant engineering, actually.
Yeah.
If genera represent the created kinds, then Noah took less than here's how it relates to Noah's flood, took less than 20,000 land animals on the ark. Far fewer if kinds originally gave rise to the families that we talk about today.
And so from these kinds came many daughter species, which generally each have less information and are thus more specialized than the parent population on the ark. So properly understood, adaptation by natural selection, which gets rid of information, does not involve the addition of new complex DNA information. Thus, students should not be taught that it demonstrates evolution happening as if it shows, the process by which fish could turn into, people.
Right. Understanding what God has told us in Genesis provides a solid foundation for thinking about the classification of living things and how the great diversity we see around us today has come about.
Right. So when we get back, we're going to talk about a specific example that support what we've been talking about here.
Evolutionists have long proposed that nearly all our DNA, 98%, is junk, that it has no function whatsoever. They claimed this because evolution needs lots of junk DNA for three reasons. Firstly, there are far too many mutations which would cause our extinction, but if nearly all of them occurred in junk DNA, then they are less of a problem. Secondly, if mutations, accidental changes, created us, then accidents cannot create DNA with 100% function. This would be unbelievable. Thirdly, if mutations created us, they must have occurred in lots of DNA that had no function to allow lots of experimentation without damaging the existing functions. However, modern science reveals that nearly all our DNA has a function, and this is a huge problem for evolution. Creationist scientists doubted the junk DNA idea all along, although because we live in a fallen world, they expected to find some damaged DNA that might appear to be junk, but not much.
To find out more from Creation Ministries International, visit our website, creation.com.
Welcome. Our subject this week is how does genetics point to design? Now, we've explained that the creation model says that rather than simple creatures with limited amounts of genetic information being built up slowly with more and more sophisticated variants over millions of years, rather God started creatures out with an amazing amount of variability, genetic information there, so that they could adapt to whatever environments they encountered down the road. We've got a specific example here to show just that. The multiple genomes of Arabidopsis.
Right. Now, in 2007, Science reported on the genome of Arabidopsis thalania, a flowering plant of the mustard family with a small genome that's suitable for extensive genetic studies. And this report was of particular interest because it showed the genomes of 19 individual plants collected from 19 individual strands, ranging from subarctic regions to the tropics.
Okay. Now, according to a commentary summarizing the results of this painstaking analysis, about 4% of the reference genome either looks very different in the wild varieties or cannot be found at all. Almost every 10th gene was so defective that it could not fulfill its normal function anymore. Results such as these raise fundamental questions. For one, they qualify the value of the model genome sequenced so far. There isn't such a thing as a genome of a species, says Weigel. He adds, the insight that the DNA sequence of a single individual is by far not sufficient to understand the genetic potential of a species also fuels current efforts in human genetics.
Now that's amazing, because what he's saying is, look, there is no such thing as a genome of a single species. What they're finding is all over the world, the same plants are adapted to different environments, but the degraded remains of genetic information that could allow it to survive in a different environment remains in there. But because it's not being used, mutations accumulate and it's just degrading. But it originally did have the information, so that plant could have been here and this plant could have been there. It's just amazing. And continuing on, they even said this, that even in a minimal genome, every 10th gene is dispensable, and that's been a great surprise, admits Weigel.
Wow. Now, among the 19 strands of Arabidopsis, we find there's dramatic differences. We observe genetic losses as well as genetic novelties, although the dispensability of genes is easy to understand with respect to genetic redundancy. The observed novelties are much harder to conceive unless we accept that all observed novelties are not novelties at all, but genetic tools that have resided in the genome since the day Arabidopsis was created. The genetic novelties may simply reflect environmental constraints that have helped preserve these genetic tools.
Yeah. There is indeed no such thing as a genome of a species, because what we observe today are the rearranged and adapted genomes that were all derived from an original genome that contained all the genetic tools we find scattered throughout the population. The great surprise is only a great surprise with respect to the Darwinian paradigm, right? So with a pluripotent Arabidopsis genome in mind, the data is not surprising at all. It's in accord with what we might expect from the perspective of a rapid repopulation of the Earth after the flood.
Yeah, there we go. Modern Arabidopsis genomes look as if they were derived from much larger genomes containing an excess genetic all kinds of excess genetic elements, both coding and non-coding, so repetitive sequences that can easily be lost, shuffled, or duplicated. The dispensable genes outlined here can be understood as genetic redundancies originally present in the genomes that over time steadily fell apart, as you mentioned there, in those 19 individuals, because the environment didn't select for them.
Exactly. So the study strongly suggests that isolated strands of plants originated as a result of loss of genetic redundancies, duplication and rearrangement of genetic elements, and the dispensability of the of the genes of Arabidopsis could have been predicted because most of the genes still present in the individual genome are redundant. These observations strongly favor this abundant genome hypothesis, if you will, that creation has put forward.
Yeah, so known examples of rapid speciation in modern times are in perfect accord with the Bible. Just variation within the created kinds. All known examples of modern day speciation involve a loss or reshuffling of existing genetic information.
Right, and if speciation is not evidence for evolution, reversing it obviously has nothing to do with undoing evolution.
Right,
if all it takes to cause two species to become one is the reshuffling of genes, then a gene reshuffle presumably causes the original original species to, split into isolated species. And since this involves no new information, it can't legitimately be used as evidence that yeast can become yaks, if you give it enough time. And we'll be back in just one moment.
The vigorous promotion of evolution as established fact is causing many Christians to question the biblical creation account, and some non-Christians won't consider Christianity because they believe the Bible has been disproved by science. That's where Creation Magazine comes in. Creation Magazine is a family-friendly publication packed with cutting-edge science that supports the Bible, presented in an easy-to-understand format by some of the leading experts in their fields of study. Visit creation.com to subscribe today.
Well, welcome back. This is the In the News section here on Creation Magazine Live, and this article we're about to read from, it's called Scientists Observe Wasps Evolving into New Species. And this is going to relate directly to what we were talking about earlier, and it also speaks to, once again, how in the news and so often people are exposed to the concept of evolution, saying, look, we're observing evolution. Now, so often you see these conflicting reports. We've observed evolution. Evolution hasn't been observed. So this just goes to show that what they're talking about here is speciation involving natural selection, genetic mutation, but not evolution.
Which we agree with. We've always agreed with. We write papers on, and a lot more papers recently on that.
So this isn't observing evolution.
Not evolution at all.
So they say scientists have documented 3 species of wasps turning into 3 new species, an unusual close-up view of rapid evolution in action. In a study published in the Proceeding of National Academy of Sciences, the team found that an evolutionary changes in one species of fruit fly triggered a cascade of evolutionary changes in 3 species of wasp that are predators of the fruit fly. As a result, not only did the fruit fly evolve into new species, but so did each of the wasps. The speciation process took a mere 160 years. The research sheds light not only on how some new life forms originate, but also how quickly it can happen. We tend to think of evolution occurring over millions of years, says Scott Egan, evolutionary biologist at Rice University and co-author of the study. What jumped out at me is that a new species can emerge in contemporary time.
Yeah, so let's just stop there and comment a little bit.
Right.
They're surprised that speciation can happen quickly. Not a surprise to us at all. In fact, it's predicted by the creation model.
Right, and even old Earth creationists have accused young Earth creationists of believing in more rapid evolution than even the evolutionists do, because they say if you went from two of every 7 of some creatures on the ark, and now we've got all these different species, you guys believe in rapid evolution. Well, number one, speciation is not evolution.
It's not evolution. There's this terminology. We needed properly defined terms. And the folks in this article, they're talking about, this is an unusually close-up view of rapid evolution in action. It's rapid speciation.
Speciation. But that's not evolution. These wasps didn't turn into anything other than wasps, and these fruit flies didn't turn into anything other than fruit flies. It's a reshuffling, a recombination of genetic material, exactly what we were explaining earlier on the show. This isn't evolution in action.
Let's continue. Speciation typically occurs when biological variations between individuals in a species allow some of them to live in a different environment. If they thrive there, it drives divergence from the original pool and a new species is born. But such speciation in one group of organisms also triggers speciations in another group of organisms? But can such speciation? The latest study is among the first to document that it does. So that is fascinating.
Yeah, and once again, it just shows that science actually supports the creation model. And we'll see you next time on Creation Magazine Live.